Good.
Recording in progress, it says. That's marvellous. Marvellous. We got the technology together. It only took me all day. Welcome, one and all. Look, this is going to be something a little bit different. I haven't done this before. What we're going to do is do a look at a research paper that I've been asked to comment on and to help Marcus understand. Hello, Marcus, by the way. Good to see you.
Yeah, hello.
I enjoyed our discussion the other day, and I thought, well, let's have this young man back, because I'm all about supporting the young people and trying to educate and encourage young people to say the right things, do the right things, have the right mindset, all of that. So hopefully this all works and it all records the way it should, and the recording comes out with the paper up on the screen. I'm a bit of a technophobe, so there it is. So what I'll do is I shall minimize the window of you and I for us, and hopefully there's a nice big version of the paper up on the screen. So Marcus, tell us about this paper as far as you understand it. What do you think it is? What do you think it basically says in a nutshell? What is it that grabbed your attention on this paper, and why did you bring this paper to me and say, tell me about this paper? There you go. There's a lot, but...
I heard that it was one of the major papers that kind of introduced the lipid hypothesis and kind of solidified it in nutrition dogma. Okay. And what I think it is testing fatty streaks in the artery in correlation to the LDL lipoprotein levels in each person.
Right. Okay. Okay. Very good. So it sounds like what you're saying is that several commentators who hold a view antithetical to your view around the non-issue of cholesterol and heart disease have put this paper in front of you and said, nah, here's a paper that absolutely proves my point and shows that you're an idiot, Marcus.
Essentially.
And what do you think of that? Okay, right. Well, let's deal with it. Let's do that. All right. So hopefully, as I say, hopefully people can see the paper up on the screen. It's from the New England Journal of Medicine in January of 1986. And it says, well, the title says, Relation of Serum Lipoprotein Levels and Systolic Blood Pressure to Early Atherosclerosis. In other words, the study known as the Bogalusa Heart Study. Okay. So straight away, there's some clues. When you're reading a paper, you can read the title of a paper and see what kind of paper it is, usually, if the title is at all reasonably written. Mm-hmm. This one is in that the very first word in the title tells you what kind of paper this is.
Okay.
Relation.
Yeah. This
is related to that. So this paper is going to tell us when we read the thing.
Yeah.
Okay. So let's say that everything in this paper is absolutely rock solid, and let's say that serum lipoprotein levels, whatever that means, and systolic blood pressure, however that's measured, is related in some way to early atherosclerosis, whatever that is. Let's say all of those things are true. So what?
Yeah.
Sunburn is related to the sale of ice creams. Does that mean we deal with the sunburn scourge by outlawing the sale of ice creams?
Yeah. No.
No, we don't. No. Shark attacks are related to ice cream sales. This ice cream stuff's dangerous, isn't it? I'll tell you what else is related to what else. Fire crews are highly and predictably related to forest fires. And house fires in general, in fact. I'll tell you what else is related to what else.
Yeah.
Drownings in swimming pools is highly and predictably related to the release of movies starring Nicolas Cage. I think that's probably causal.
Yeah.
That one.
It sounds like it, yeah.
Because Nicolas Cage is just awful, isn't he? All right, so straight away we know the value of this paper in terms of talking or asserting in an argument anything whatsoever to do with cause or effect. is this a paper which experimentally looks at the relationship between factor X or factors X, Y, and Z versus an outcome variable of some kind in an experimental protocol under proper discipline control, sample sizing, randomization, all the things that we have to do under the accepted epistemology of science to be able to at all assert anything whatsoever about cause or effect. No, it's not that sort of paper.
Right.
So when somebody puts this paper in front of you and says, look, this proves my point, the correct and appropriate response is, the hell it does. No, it does not. Before we've even got beyond reading the first word in the title of the paper. We're done. You win that argument.
All right.
The rest of the paper's a waste of time. And it really is when you read it. But we'll get to that during the discussion. So that's the first point for people to understand. When you are presented a paper which purports or is purported by the person waving that paper in front of you to be absolute proof of cause and effect in any way, shape, or form, question one is, okay, is this paper an experimental piece of work conducted under proper control, proper discipline of observation with proper randomization, adequate sample size for power, etc., under the accepted epistemology of science as that regards cause and effect? That's your question. To which the person that's waving that paper in front of you will either have to say one of two things. No, it isn't. Well, they'll have to answer correctly, no, it isn't that kind of study. Or they'll have to answer, I don't know. In which case, if it's the second response, I don't know, then you can jump all over them. You can say, well, that being the case, what in a purple fit makes you think you are remotely competent to wave this paper in front of me and tell me that it proves something? You don't even know what kind of paper it is.
Yeah.
And then you can tell them, well, the clue is in the first word of the title. This is a correlation study. This is correlated with that. We're done. You win the argument. Try again. Have you got any other references? Please provide us with a cause and effect paper if you want to make a cause and effect statement.
Yeah, right.
This causes that? Great. Well, show me that that's so.
Would you be able to explain how you go about determining, whether it's gone through proper methodology of
well, absolutely. We can do that. We can do that. Yes. Okay. So an experimental study, if you want to make rock solid statements or even reasonably good statements around cause and effect, what needs to happen is you need two subpopulations who are in every respect, prior to the experimental protocol, identical. Now that's real easy if you're talking about how does a sodium atom respond to a chemical reaction with a chloride, for example, a sodium ion, I should say, and a chloride ion.
Okay.
That's predictable. All sodium atoms are pretty much identical with very minor changes, and the same is true of chlorides. That's easy. All we need now do is observe this relationship occurring on enough occasions to be absolutely certain that it wasn't a one-off that happened that was just out of the box. And we can state with absolute certainty, therefore, that we have a cause and effect relationship here. We know what happens when you mix sodium and chloride together, because there are very few degrees of freedom. Make sense?
Yeah.
Not true of a human body, is it?
No.
Vastly complex organism construed of trillions, with a T, trillions of individual cells, all working individually but in concert according to the overall machinations of the homeostatic mechanism. There's some big words for you. In such a way as that any one of a myriad of uncontrolled degrees of freedom could well be affecting the outcome in such a way as to invalidate any claim around the so-called causal agent.
Yeah.
Ergo, you need two populations. If you want to talk about the effects of X, Y, or Z on a human body, you need two populations of humans, not mice or rats or pigs or dogs or anything else, monkeys, humans, okay? Because that's the rules.
Yeah, of course.
So you need two populations of humans. They need to be identical in every respect before you start, i.e. genetically identical. So we need two sets of twins. Twin A in group A and twin B in group B, and the same for every one of the twins.
Right.
Okay? So straight away, this is not likely to occur.
No.
Secondly, they need to have been treated identically since conception, these twins. Because anything that happens to those twins prior to the experimental protocol could have an effect on the outcome. Could it not?
Yeah.
Yes.
It definitely could, yeah.
So it's looking pretty poor in terms of, are there any studies that can reliably inform us on cause or effect?
Right, yeah.
With an absolute certainty. Next thing, not only do we need sets of twins that are identical and have been treated identically prior to, there also need to be enough of them so that we have statistical power sufficient to say that our result is unlikely to have occurred due to dumb chance alone. I know that's a parsimonious description of the thing. Bear with me, those of you that have some statistical knowledge. It's near enough. Okay? So we need statistical power, so we need a great big, probably, sample of sets of identical twins.
Yeah.
So it's looking even less likely that we'll be able to recruit that. We need to apply which twin gets to end up in which group in a truly randomized way. Because if you put all the oldest twins, because twins aren't born at the same time, so you put the oldest twins in one group and the youngest twins in the other group, you have got two random groups. So you need a method to, and that's true of any other degree of freedom that's, different between the twins. Next thing, if you want to make a statement about the effect of, let's say, serum lipoproteins and systolic blood pressure on early atherosclerosis, for example, well, you need to keep those people under lock and key, observation and control long enough to know that nothing else could possibly have intervened and caused this issue to occur. So you're not going to get that past an ethics committee.
No.
You're not going to have the finances to do it because that's going to be a years and years and years long study with people locked in a laboratory. And if you do all of those things correctly, absolutely correctly, now you have a study that's absolutely internally correct, valid, right? However, what you do not have in that case, that you've done all of those things correctly somehow, what you do not have is a result that is externally valid to any other population.
Yeah, right.
So what I'm telling you in a nutshell is, in terms of cause and effect, input leading to health outcome of some kind, the exact amount of science that is available in the literature regarding human beings that you can read on any topic regarding health outcomes is not one scrap.
Yeah.
Not a jot. Not a single paper.
Yeah, right. Okay.
So it doesn't matter what paper they wave in front of you. You can say, sorry, nope. What you have there is a paper that makes an inference.
I guess so. Yeah.
It's a paper that makes an inference. It doesn't prove anything. It infers something might be so.
Okay, right.
Now all that's needed is for a sensible and educated discussion about how good that inference might be.
Yeah.
I.e., what is its veracity? What is its utility? Should we take that inference seriously and adopt it in our lives? Or should we shake our heads at these ridiculous buffoons that call themselves the vegans and go, you clown, you scientifically illiterate buffoon, are you serious? And I suggest the second course of action, because they are imbeciles.
Right.
Right.
Yeah.
So there it is. That's the appropriate. And a lot of people, when I sort of have that kind of discussion, when we kind of teach that, when I teach that, a lot of people, even academics, not that being an academic makes you of any veracity or correctness, it just means you're an academic, right? But even academics often say to me, so you're a science denier. That's what they say to me.
Wow, really?
Right. So what? My response, and my response to that is immediate and straight away. It's easy. My response is, no.
It's the opposite.
It's the exact opposite, you imbecile. I am insisting upon science.
Yeah.
I'm not denying it. What I'm denying is the stuff you're telling me is science. I'm denying that is science. It's not. It's theology. It's associative mishmash. It's epididdly-doodly-mology. It's poorly controlled, poorly randomized, underpowered claptrap being passed off as cause and effect inference when it's nowhere close.
Yeah.
Sorry if that's, challenging to you. Not sorry. Those are the facts.
Yeah.
I'm the science me. I'm safe and effective. Trust me, Marcus. Never question the science.
No, never.
Good. All right. So let's dive into this paper a bit now that we've actually destroyed it before we even look at it. The first word destroyed it.
Okay.
Look, it's a correlation study. We're done. Unless you want to tell us this is related to that, in which case, if the person that you're arguing with is smart enough not to claim it's cause and effect, but to say to you, well, look, these two things are related, or these factors are related, here's my next question then. In that case, I say to that person, awesome, there's a relationship. Please provide the R-squared value and the 95% confidence interval around that R-squared value.
Okay. Can you explain what that means?
Exactly. Right. And that's what they'll say. In which case you can say, well, that being the case, it's clear you don't understand the statistical inference made by this paper, what it means, how to interpret it. Ergo, why are you waving this paper in front of me? What makes you think you're qualified to do that?
Yeah.
Richard Burgess. You imbecile. Oh, my word. But what do I know? I can't bench 415 pounds.
Oh, yeah. He always brings up the bench press.
As it turns out, neither can he. Anyway, that's for another day. Right. So, where were we going? We were going to read the paper a bit. That's what we were going to do. Yeah. Oh, I was going to tell you about the R-squared thing. That's what it was. You asked me a question. Yeah. Okay. So when you take variable X, which is your so-called input variable or your independent variable, that is proposed to have a relationship with your so-called dependent variable, the y-axis. So you basically have an XY plot.
Okay.
Then you take the data points of your individual subjects in your subject pool and you plot them on that XY chart according to what the value of the independent variable is. Again, let's ignore the error of measurement for a moment, just to say all measurements are absolutely accurate, which they're not.
Right.
And then we plot them against where they would sit on the Y variable. Again, error of measurement, but again, let's just pretend that doesn't exist, even though it does. And you get a scatter plot, or you get a bunch of dots on a chart.
Yeah.
If X is related to Y in a systemic way, you'll be able to draw a line through that data set in such a way as that most of the data points are very close to the line, and very few of them are not close to the line. And that line might be a positive straight line. It might be a negative straight line in terms of its slope. Or we can get really complex. It might be curvilinear according to a first, second, third, fourth, or umpteenth order polynomial curve.
Okay.
How do we determine the best line? How do we determine the line of best fit? Well, there's some math that can do it to help us work out the least residual.
I remember that from 10th grade.
Or we can ask Excel spreadsheet to do it for us.
Okay.
And tell us what line is best fit.
Yeah.
Then what you get is you get a mathematical description of the slope of that line or the curve of that line, as the case may be. Plus you get an indication of the error around that line.
Yeah. Okay.
And the indication of error around that line is a thing called R.
Okay.
Pearson product number. And it's a number that ranges between 1.0 and minus 1.0, where 1.0 is a perfect positive correlation, negative 1.0 is a perfect negative correlation, and 0 is no correlation whatsoever between these two factors. So you can say X is related to Y according to this slope line here, where it says X is equal to Y plus a factor with an error around it, R squared equals. And R squared is the R value multiplied by itself.
Okay.
Right? And the R squared value is thought of as the coefficient of causality. It still isn't, but that's just the title of the thing.
Yeah.
What it is it tells us the shared variance between X and Y. So as X varies, so does Y. You can think of the R squared value as, if you multiply it by 100, that's the percentage of shared variance between X and Y. So if your R squared value is 0.36, that means 36% of the variance between X and Y is shared.
Okay, right.
And 60, whatever it is, up to 100, the rest of it, 64, of course, is not shared. Ergo, what's the utility of that relationship? If 64% of the variance is not due to the thing that you're saying is the cause.
Yeah, that's not at all.
Sounds like you haven't got the picture covered. You should probably go back to the drawing board, shouldn't you?
Yeah, probably.
So that's the question. When they say, if they don't say this is cause and effect inference, most people will just say this is cause and effect because they think that's what science is doing. The more educated will say, well, look, this isn't a cause and effect thing. This isn't a proper experiment. This is an associative study, and it shows that these things are related. So then you say, well, great. Please provide me with the slope of the line and the R squared value around that anytime you like. And they'll then say, what? Hey, what? In which case you can say, well, why the hell are you telling me that you know more about this than I do?
Yeah.
You don't understand what the inference is here. You don't know what an R squared value is. We're done. You lose. I win. Thank you very much for coming. Thanks for the money. If you want. Okay. So let's read through the abstract of this paper, which was done by a bunch of people whose names you can see on the screen if everything's working the way it should be working.
Yeah.
It says abstract, which means a precise short summary of this paper, what it does, what it says, what was investigated, and what the outcome stats were. You can generally tell the quality of a paper by reading the abstract, and if the abstract meets certain requirements, you can go, this is probably a reasonable paper. And if it doesn't, you can go, well, if the abstract is nonsense, then the paper itself is not likely to be anything but nonsense, is it?
Right. Okay.
And there are things like, is this a clear and concise description of exactly what was done and a summary of the outcome statistics that came out of that with a very brief sentence or so at the end, which says, therefore, the inference appears to be.
Okay.
All right. So here we go. Let's see if it meets that. Am I boring you?
No, I'm tired.
The young
You're
people.
not boring me. You're not boring me. You're not boring me.
I'm kidding, Marcus. I'm just teasing you.
Of course.
Yeah. All right. So, abstract. We assessed the relation of risk factors. Okay, let's stop right there. Risk factors, it says.
Right, which they didn't do.
They did not do that.
No.
Risk is a cause and effect statement necessarily. Necessarily, it's cause and effect.
Yeah.
Because if you say this is risky in some way, great, what's the mechanism of that risk?
Yeah.
They did not do that.
No.
So they are misrepresenting their study in the first sentence of the abstract.
Yeah, right. Okay.
So it's likely to be a paper that's problematic in terms of the overshoot of its authors, in terms of their failure to apply the correct disciplines of how to communicate about what it is they have actually done.
Right.
Anyway, risk factors. What they mean by risk factors is their proposed independent variables that they are proposing are related in some way to their outcome variable, So let's just read it as that. It is not a risk factor. It is an independent variable or set of independent variables, and that's how it should be described. Right. Sure. We assess the relation of risk factors for cardiovascular disease to early atherosclerotic lesions in the aorta and coronary arteries in 35 persons. We're done. If we weren't already done, we're done. 35 people. N equals 35. Okay, so what can we deduce about 7 or 8 billion different people on the basis of observing in some way 35? Very little.
Yeah.
Very little indeed. So the statistical power of this study is likely to be substandard.
Yeah.
And I would suggest that this paper does not discuss or describe any calculations around statistical power that they may have made. And I would go further to suggest that it's unlikely they even made any attempt to assess the statistical power of their inference.
Right. Okay.
So we're done. If we weren't already, we are already done in the first word. It's a relationship, right? But if that didn't do it for us, now we're done. 35 persons. But let's carry on, because in brackets, right after that, it says mean age at death, 18. 18. Okay. 35 people who died at an unnaturally young age for a person to die. For some reason. Are they representative of a wider population to which we can then extrapolate this finding? No.
Definitely not.
Absolutely not.
Yeah.
Now we're done, if we weren't already, which we were already.
Yeah.
We're done again. We're double done. Done. Yeah. All right. Good. Okay. Let's carry on, though, because we've started, so we might as well completely put this thing to bed. Aortic involvement with fatty streaks. What the purple does that mean? Do you see how that's just words?
Yeah.
Aortic involvement with fatty streaks. Well, they're in a relationship together, or they have some love affair, or what?
Yeah, I couldn't tell you what that means.
No, exactly. So that's poorly written. Anyway, aortic involvement with fatty streaks. P is less than 0.01. Okay. So what they're saying is 35 persons who died at an unnaturally young age of 18 for some reason, whatever it was. And what they found was that of those 35 people, very few in the first place, those of them who were black had a different prevalence of the thing they're looking at, the fatty streaks, whatever that means, than whites.
So it's completely different populations that they're using.
Well, let's think about that for a second.
Genetic.
Let's think about that for a second, shall we? If I tell you, Marcus, that I am a black man, what can you say about that?
That I don't know anything about how you would respond to different things or how you work.
Am I a black man, Marcus?
Oh, no.
How do you know?
Because I can see that you're not.
Oh, so what would be different if I was a black man?
All of your genetic makeup.
How do you know? Have you tested my genes?
Your genes? No.
No. So if I tell you I'm a black man, Marcus, who are you to say I'm not, you bigot? The thing that would be different is my skin would look a different color than what it looks, and some of my features might look a bit different, and maybe I'd be taller than 5'6". Maybe I'd still have hair.
Maybe
my penis would be larger. I'm told that's the case.
Yeah.
You know?
Yeah.
Maybe it wouldn't. Who knows? Let's not give away too many secrets. Wink, wink, nudge, nudge. Say no more. So how do you determine who's a black man and who isn't?
By
genetic. Okay, well, you've mentioned it. Test the genes. Okay, great. What genes do black people have that white people don't?
Yeah, I don't know. I mean, I do know that DNA testing is very subjective in
correct.
Ancestral.
The entire gambit of gene sets across the human species. Well, we share 99.9 something% of our genes identically.
Yeah. Okay.
So good luck taking taking some samples from some people, taking those blinded samples to a lab and asking the lab technicians to do some tests and tell us which of those people are black people and which are not.
Yeah, you're right. It's all bullshit.
Assuming that there is such a thing as you're either black or you're not. Because what happens if your mum was black and your dad wasn't, whatever that means?
Yeah.
You'd be 50% then. What if neither one of them were 100% before that? Well, then we don't even know, do we?
True.
So I think this paper is starting to look a bit shaky. If it wasn't already. Anyway, whites versus blacks, it says. So 37 versus 17%. So in the people that somehow are identified as black, 37% of them versus 17% of those 18-year-olds in those two separate groups showed fatty streaks. So why would you bring that forward as the most prevalent finding of the study, as the first thing you talk about in the abstract? Because it says, the title of the paper says, the relationship of serum lipoprotein levels and systolic blood pressure to early atherosclerosis. Yeah. It doesn't mention anything about being black or white in the title of the paper.
Yeah.
Neither does it say it's going to make a comparison between black people and white people. Neither does it mention that we're only talking about 18-year-olds on average who are not representative of the population, in that most 18-year-olds on average aren't dead.
Yeah, most of them are alive. Yeah, of course.
Dear, oh dear. Oh, for what it's worth, that's what they found. Whatever that means. It means nothing. It proves nothing. It shows nothing, except the arrogance and ignorance of the people who wrote this paper.
Yeah.
Their lack of discipline, their lack of thought, their lack of comprehension of objective reality. And if that's the case, should we take anything that's written in this paper remotely seriously from that point forward? Because this is supposed to be a science paper.
Seems not.
It seems not, doesn't it? All right. Let's carry on, though, because we started. So, whatever. All right. However, it says, aortic fatty streaks, whatever that means, were strongly related to ante-mortem levels of both total and low-density lipoprotein cholesterol. And then it gives an R value. Hurrah! And a probability statement around the confidence of that R value.
Okay.
However, there are several problems with this sentence, because there are with just about every sentence written by just about every author and just about every paper, it seems. Shall we go over those?
Sure.
1, aortic fatty streaks, it says. Well, I presume they'll define what the hell that is in the paper, because it's not done here in the abstract, so we don't know whether or not they're even looking at a valid output statistic at all. We just have to assume at this point that they are.
Yeah.
All right. Strongly related, it says. Well, that's a value judgment statement. See how they've given it says after, in brackets here, it says R is equal to 0.67.
Yeah.
P is less than 0.0001. Okay, well, that's the information. We do not need a value judgment statement strongly.
Yeah, true.
Strongly is a value judgment statement. It's an opinion.
Yeah.
Okay. Opinions, Marcus, are just like anal orifices. Sorry, anal orifi is the plural. How are opinions just like anal orifi? Well, almost everybody's got at least one, and it's full of shit.
Right.
That's what an opinion is.
Yeah.
R equals 0.67, P is less than 0.001 for this small population of unnaturally dead young people is not an opinion. It's a statement of statistical finding around the correlation between X and Y, whatever X is and whatever Y is. All right. That said, R squared that we spoke about before.
Yeah.
If you take 0.67 and multiply it by itself, you get 0.3 something, don't you?
Yeah, about that.
So 30% or so of the variance between X and Y, whatever the hell those are, is shared. 60-something% is not. So who cares
so it's
how
very
likely it is to be statistically,
Yeah.
It's weak. It's piss weak at best.
Yeah. Okay.
Absolutely piss. And they've said strongly related. No, it isn't. It's piss weak.
Yeah.
The numbers right there. We can read the numbers.
Yeah.
Those of us who know that what statistics are can look at that straight away and go, no, not strongly related at all, in fact. And given that n equals 35, are you stoned? These authors? This is nuts.
Yeah, okay.
All right.
Yeah.
Good. So, anyways, 0.67 is the R value, so 0.3 something R squared. P is less than 0.0001, which means the chance of this particular level of correlation having occurred due to dumb chance alone is pretty low. So what? For each association. Yeah. Okay, well, good. Independently of race, well, hang on. Is race a thing or is it not? Sex, by which they mean gender.
Yeah.
So are there two options for that or 15? This is 1986, so I suggest probably two.
It's two.
Yeah, and two. It was back in 1986. There were two genders. I don't know about 2026. How many genders there are?
Well, if they said sex in a study, wouldn't they would be referring to the male or female, even in nowadays, wouldn't they?
Are you a bigot, Marcus?
Maybe.
In 2026, you are. Sex, you say, meaning gender, male or female. Goodness, no, there are umpteen different genders, are there not?
Maybe, I don't know. Maybe I'm a bigot.
And age. Well, it says independently of age. Well, hang on.
You
haven't age limited
to 18. They all died at 18
on average.
Yeah.
Again, it says they died at 18 on average, but doesn't give us the spread around that.
No.
Wow. This is good science, though, Marcus. I'm glad this person brought you this paper and said, look, this is science that proves my point. I'm happy that they proved you wrong by showing you this. All right. And we're inversely correlated with the ratio of high-density lipoprotein cholesterol. Well, there's the next problem. There is no such thing as high-density or low-density lipoprotein cholesterol.
Yeah, it's just the lipoproteins itself.
That's right. Yeah. Cholesterol is cholesterol. It only comes in one form in the human body. There's only one isomer of it. And there's no good ones and bad ones. Therefore, they're all the same.
Yeah.
And to suggest that the kind of lipoprotein tells us what kind of cholesterol it is laughable. If I'm a bald-headed bigot from New Zealand and I drive my Subaru Impreza one day, does that make me a different person the next day if I take out the Hi-Ace van instead? No. Cholesterol is still cholesterol. It doesn't matter what's been carried by.
Yeah.
And if you're talking about low-density or high-density lipoprotein cholesterol, what is it that you're actually measuring? Is it the lipoprotein or is it the cholesterol carried by it?
I can assume it's the lipoprotein.
Well, you might assume that. That might be a reasonable assumption that they've left you to make. Very scientific.
Yeah.
However, you'd be wrong again. Because when they talk about low-density lipoprotein cholesterol, the method by which they measure it in a laboratory, Marcus, is they don't. I know, that stumbled. Richard Burgess fell over that one too. Well, yes, they did, Bart. They measured it.
Yeah.
No, Richard, they didn't. But they did. No, Richard, they still didn't.
Yeah.
But they must have. You'd think so, wouldn't you, Richard? But you'd be wrong. They didn't. They estimate it.
Okay.
That's not a measurement, is it?
No.
It's an estimation. And in my estimation, Richard Burgess, you're an imbecile.
Yeah.
I think I said words. Some of the words I used on that day were slightly shorter words than the ones I'm using with you, Marcus. Mostly because you're not an imbecile and because you have the capacity to understand words that have more than 4 letters in them.
Well, thank you.
That's right.
And Richard doesn't?
No.
Right.
In fact, if I labelled his elbow and his anal orifice and taught him to read, he probably still couldn't determine which was which.
Yeah.
Yeah. Anyways, there it is. Right. So we've got a bunch of correlations between the associations there with various different subfractions of the lipoproteins, so-called. Some of them measured, some of them estimated, so invalid from the ground up. But let's read through those just for fun. So high-density lipoprotein cholesterol, which does not exist, to low-density plus very low-density lipoprotein cholesterol. Hang on, which? What? Which one?
Yeah.
What does that sentence even say?
The ratio.
Let's go back to the beginning of the sentence because I've been, diving in and out of the actual verbiage in the paper. But let's go back to it just to see how much sense in common English this sentence makes here.
Yeah.
Okay. All right. Let's go back to the beginning of it because it's quite a long sentence, which is poor, by the way.
It is. Yeah.
It says, however, aortic fatty streaks were strongly related to ante-mortem before death. Ante-mortem levels of both total and low-density lipoprotein cholesterol R equals 0.67, P is less than 0.001 for each association, independently of race, sex, and age, and were inversely correlated with the ratio of high-density lipoprotein cholesterol to low-density plus very low-density lipoprotein cholesterol. R equals 0.035, negative 0.35, that is. P is 0.06. Okay, number one, very long sentence, way too many words, unclear what the subject of the sentence is, what was being measured, and what the outcome is that we should take from that at all. Very poorly written indeed.
Yeah.
Secondly, the epistemology of science, rightly or wrongly, and it is wrongly, but nonetheless this is still a rule, is that you make a dichotomous decision that a thing is a real effect, likely to be occurring due to the experimental procedure and unlikely to be occurring due to dumb chance when you meet a threshold value of the P value.
Okay.
In most studies, that threshold is 0.05.
Right. Okay.
Or below. This one says P is equal to 0.06.
Yeah.
Ergo, that data should be rejected.
Yeah.
As not entirely uncertain to have occurred due to dumb chance.
Yeah.
Now, when we think about it, we have a completely non-representative population of individuals who have unnaturally died at a very young age, without telling us what the spread around that average was at all, numbering 35 in total. I wonder if dumb chance had anything to do with any of this. Looks like it did.
Yeah.
It is equal to 0.06. I think we're done. If we weren't already done, diddly done, diddly done.
Triple done.
Done, diddly done, done. Right. Right. So let's carry on anyway, because why not? Coronary artery fatty streaks were correlated with very low density lipoprotein cholesterol, which still doesn't exist. R is equal to 0.41. P is equal to 0.04. Okay. Mean systolic blood pressure levels also tended to be higher in the 4 subjects with coronary artery fibrous plaques. Okay. So tell me this, Marcus. Now we're looking at X and Y.
Yeah.
2 independent variables. We're looking at some aspect of the lipids and some physiological signal like their blood pressure.
Yeah.
And talking about the relationship of those 2 things with an outcome which they still haven't defined.
Yeah.
Okay. Let's say one of those things is causal. Given that they've measured 2 things and found relationships between 2 things of whatever level and the outcome thing, whatever that is, which one was the causer?
Can't tell.
That's right. You cannot tell. That's exactly what I mean by uncontrolled degrees of freedom.
Right.
Let's say they were both causal to some degree. How much was each one causal? what was the proportion of their contribution?
Yeah.
And before those of you that know something about this say, oh, we can do multivariate regression, you need to watch more of my videos. No, you can't. That's fabrication. Scientists report what they observed. They don't take observations and adjust the observations to something that they did not observe. That's fabrication.
Yeah.
That's not control, because you can't exert control after the fact. Control has to be exerted before you collect the data.
Yeah. Okay. Yeah.
Dun diddly dun diddly dun diddly dun dun dun. I think we're really getting very, very close to being very, very done if we weren't already. Okay. So mean systolic blood pressure levels also tended to be higher in the four subjects with coronary artery fibrous plaques than in those without them.
In the 4 subjects. Wow.
Correct, the 4. And P is equal to 0.09. So was that, in fact, something that tended to be the case?
Yeah.
No.
No.
It wasn't. According to the rules of the epistemology, the dichotomous decision no tree. I'm not saying I agree with that being a procedure. I think exact P values should be given at all times and all data should be presented. So I congratulate these authors on presenting the data.
Yeah.
What I don't congratulate these authors on is failing completely to explicitly disclaim their data is of absolutely no value to anybody whatsoever.
Yeah.
None.
Yeah.
Wow. Okay, right. Here we go. And now we have the statement around how to interpret this nonsense that they've just written in the abstract.
Okay.
Here we go. These results document the importance of risk factor levels to early anatomical changes in the aorta and coronary arteries. Okay, is that statement backed up by the data they've provided?
Not at all.
Not at all. No, it isn't. No, you're right. So basically what they've said is, here's some data we collected. It's tosh. It's excremental garbage. It's pustulous, ridiculous nonsense. It's of no value to anybody. But it's very important data because we collected it. Good.
Yeah.
All right. So, no, it doesn't show that. Next sentence. The progression of fatty streaks. Hang on. Progression occurs over time, does it not?
Yeah, that's not what they even...
That's not what they even looked at. They took a one-off assessment of the level of fatty streaking and or fibrous plaquing in the aorta and coronary arteries of some deceased people.
Yeah.
Deceased people do not tend to progress in their disease process because it's done with now.
Yeah.
So this just goes from ridiculous to I don't know what. Okay. The progression, of fatty streaks to fibrous plaques is uncertain. Well, we knew that before we read this abstract. And this abstract doesn't actually tell us anything about it that we didn't know already.
No.
But these data suggest that a rational approach to the prevention of cardiovascular disease should begin early in life. Well, I agree, but you haven't told us what the approach should be.
No.
And we didn't need this data to tell us that a rational approach to intervening in regards to progressive chronic disease would be to start earlier in life. We knew that.
Yeah.
That's common sense. We don't need a paper to tell us that.
Of course.
So ostensibly what these authors are saying is we collected some data. It's absolute rubbish. It says nothing whatsoever. We're going to tell a bit of a lie that it's important data. It's not. And then we're going to make a completely non-committal statement about some common sense that everybody knows anyway, so that we can't possibly just argue with it. Well, of course.
Yeah.
Good. There you go.
Makes sense.
That's it.
Cool.
Happy with that? We don't need to read the paper.
No, not really. No.
There's no reason to read this paper. We've got everything we need right there.
Yeah, it seems so.
It's absolute garbage.
Would you be able to go through just a couple of the charts that they...
Yeah, you bet. Let's have a look.
They showed.
Because that's the other thing that people like to do when they want to cite things at you. What they want to do is usually pull a graphical chart or figure out of a paper, splash that up on the screen and say, here, look, here's your proof.
Yeah.
No, you need to read the words around the figures to understand what was done.
Right.
Had you done so, you'd have got to where Marcus and I have got to in the last little bit of time that we've been discussing this. Oh, this is nonsense. So whatever the charts say is not going to tell us anything, but let's have a look at them. Probably a good suggestion. Thank you, Marcus. Let's do that.
Thanks.
So table 1, race, okay, sex, okay, and age distribution. Here we go. Now they're going to tell us a bit about the spread in the paper. Okay, fine. Of the 35 patients examined at autopsy. All right, so age at death. The youngest was 5, it seems, and the oldest was 24.
Yeah.
All right. Nothing there about the causes of death, of course, but, whatever. And then it talks about race, black and white, because that's all there is. Yeah, all right. So that's just a descriptive table that tells us really nothing much about these people, except clearly not a representative sample of the population at large.
Yeah. And
not of a sample size which is remotely adequate to meet any kind of statistical powering of any kind of output anyway.
Yeah.
Right. Good. Okay. Table 2, comparison for mean age adjusted. There's the word you need to see, adjusted. Risk factor levels of autopsy series and reference population. Well, what's the reference population?
35.
It's got an asterisk there, so it must be, okay.
Yeah,
so the reference population consisted of 14-year-olds examined in 1973 to 1974.
Okay.
So we've taken a population of 35 people who were aged between 5 and 24 who are dead, and we're comparing those to a population of people who are exactly 14 who are not dead.
Yeah, it's completely random.
That's not apples with apples, is it? That's fruit salad.
Yeah.
So this is not a comparative population. And why would we imagine 14-year-olds to be representative of the population at large if this group of deceased people aged 5 to 24 is supposed to tell us something about something?
Yeah.
I don't know. Right. So we've got also our reference population that doesn't meet the same percentage of blackness and whiteness. So invalid. We've got total cholesterol, triglycerides, lipoprotein levels, which are all ostensibly the same statistically. There are no differences really. The HDL ratio, the same ostensibly. The blood pressure levels, ostensibly the same. And their ponderal index, the body mass index, ostensibly exactly the same. So the reference population is not different from the experimental population. And so any statement about anything whatsoever here seems to now dissolve into nothingness.
Yeah.
If it hadn't already, which it well and truly already did.
I did look at that chart and I was like, what are they even trying to say? It's exactly the same.
Yeah. Table 3, characteristics of 6 male subjects with fibrous plaques. Okay, well, n equals 6. Good. I mean, that's I could take the height in centimeters of 6 people and make a table showing what that was. Would that tell me anything about people in general?
No,
nothing at all. So that's not of any value. All right, here we have a Spearman correlation coefficient. That's the R value that we spoke about earlier.
Right. Okay.
Before we square it to get the coefficient of causality. And it talks about positive correlations through to negative correlations. The line through the middle is the zero correlation. There is no relationship to speak of here, one way or the other. There is no systemic way that any covariance occurs.
Okay.
And we have associations between the percentage of fatty streak involvement. Again, involvement.
Don't know what that means.
Exactly. What kind of involvement are we talking about here?
Yeah.
And risk factor levels. the things that we've determined to be risk factors inappropriately. So the serum total cholesterol enjoys a correlation with the outcome, whatever that is, fatty streak involvement, of around about 0.6 something, which is what we've already read in the graphics in the abstract.
Yeah.
And we've worked out that that's about a 30-something% shared variance.
Yeah.
So pretty well worthless, especially considering the very low sample size and the non-representative nature of the sample. Serum triglycerides, basically not correlated, because less than minus 0.01 is going to be around about 0.01, which is so close to zero as makes no odds on the R squared. Right. Yeah, so forget it. LDLC, what does that mean, LDLC? It refers to LDL cholesterol. Well, which is it, the LDL or the cholesterol carried by the LDL? Which thing has been measured there? Actually, and it's neither.
That's actually not measured, is it?
That's right. So that's an estimate based on a regression sum, and a regression sum has error.
Okay.
So, and anyway, even if it was really accurate, which it isn't, again, we've got about the same level of correlation, about 30-something%.
Okay. Right.
So that's not of any value. HDLC, almost not related. HDL ratio, minus 0.35. So it's going to be less than 0.01 on the R squared. Okay. Same deal for VLDLC, whatever that is. Blood pressure measurements, really nothing much to do with it at such a young age. Of course it isn't, because the blood pressure is very important, I believe, in the etiology of atherosclerotic disease development over time. But the key there is over time. And if you're dead by the time you're, 24 at maximum, that's not going to have time to have reared its ugly head. Right. So that's not a surprise. And the body mass index had really nothing much to do with it either. And then you've got a set of data which is for the coronary arteries as opposed to the aorta specifically, and that finds even less of any value or utility there whatsoever.
Okay.
Yeah. The other thing that's missing from this chart is an indication of the noise, i.e. the 95%.
Yeah, that's what I was thinking.
So this is really very, very poor indeed, and absolutely does not constitute anything remotely approaching even a reasonable inference of anything, let alone proof of any kind of anything whatsoever.
Okay. Yeah.
Right. Next figure. The thing about a figure is you should be able to look at a figure and within seconds see exactly what that figure is about and understand exactly what's on the x-axis and what's on the y-axis and exactly how that was done. You can't do that here. Figure 2, atherosclerotic fatty streak involvement. Again, what's this involvement carry on? Of the aorta related to levels of low-density lipoprotein cholesterol, which does not exist still, in 30 young persons. And then it says increasing LDLC levels are significantly related to increasing amounts of aortic fatty streaks. Let's say that was true just for fun. So what? Ice creams and shark attacks. The intake of polyunsaturated fats and marriage dissolution. Nicolas Cage movies and drownings in swimming pools. Again, I think that last one is probably causal. But I can't prove it. Also, on this chart, we've got some numbers in boxes in the histogram for some reasons. Yeah. What the hell are those numbers? Why are they there? Exactly. Yeah. You should be able to know immediately, straight away. You shouldn't have to read anything to work that out. And it says the probability of this chart happening due to dumb chance is almost none. Well, so what? The chart doesn't tell us anything.
Yeah, right. Okay.
Really? At all?
Yeah.
Especially considering that there is no definition given on this chart of what the hell is fatty streak involvement.
Right. Yeah.
It says percentage or percent of total surface involvement. The total percentage surface of the inner space there, the endothelial beds, I suppose, that we're left to assume that's what it means.
I guess so.
However, you might have mild fatty streaking over 100%, and that's of no issue. You might have severe fatty streaking over 10%, and that might be a real issue.
Yeah.
In terms of the actual clinical meaning and outcome of that.
Right.
So this chart tells us absolutely nothing of any value either. Dear oh dear. What do we have here? Table 4, associations between percentage of total surface involvement with fatty streaks and selected cholesterol-related, again, risk factors, according to race and sex again. Oh, well, we've done it with that.
Yeah.
Table 5, age-adjusted levels. As soon as you see the word adjusted on any data set... What could that mean? Well, I'll tell you. As soon as you see the word adjusted on any data set presented anywhere to you in the literature, you know that what you're looking at is a set of data that has been fabricated.
Right. Okay.
This does not represent an observation made. They've taken the observation they made and they have mathematically changed it using a thing called multiple variate regression to a different value.
Okay.
A value that was not observed. The first assumption of a multivariate regression is that the cofactors you enter into your multivariate regression must not share any collinearity. That is the first assumption that is universally breached as soon as you adjust any data set for age of the participants.
Okay. Right.
Because we are asserting that atherosclerotic disease is a process that develops over time. As you age, you are more likely, therefore, to express atherosclerotic disease development, all else being equal, than someone who is younger than you. That is therefore collinear. You cannot make that adjustment. It is totally inappropriate to attempt to do so. It is a fabrication. It is a fantasy. It is not only pseudoscience, it is anti-science. It is academic misconduct of the highest order, born of utter statistical incompetence.
Right. Okay. Makes sense.
We're done. All right. Cool. And then we get to the references. There we go. We're done.
Cool.
There it is. This paper is excremental, postulous, worthless.
Yeah, it's utter nonsense.
Useless nonsense.
Yeah.
It establishes nothing. It infers nothing of any meaning or use to anybody. And it certainly is not an appropriate thing to throw at someone as a reference, absolutely proving they're right and you're wrong on some topic or other.
Yeah. Okay.
We're done.
Yeah.
Too easy. Good. You're welcome.
Thanks.
Stop sharing. There it is. Wasn't that fun?
That was pretty fun. Yeah, I enjoyed that.
Good. Here's what I'm going to suggest. Folks that have enjoyed this video and would like me to go through any other given papers that you have been someone's thrown at you for some reason, make an appointment on my calendar or email me if you don't have access to that, so I can give you access to it, my calendar that is. And you just need to be brave like Marcus has been and show your beautiful face on camera and just agree with everything I say, because if you don't, that's hate speech. And you will be doomed and determined immediately to be a bigot, and you will be fined and silenced. You will have to report to Lord
Sentenced
Edward Yellow,
to death,
otherwise known as Darth Correctus. and he will say things to you like, "Join me. I will complete your training. Something dark side." Okay, I'm going to go and twiddle the knobs now. Bye. Good boy. Oh, good boy. It's his one job, to twiddle the knobs.
Right.
Somebody has to go around here. It's dreadful. You can't get the help, Marcus. Right, I hope we've all learned something today. I hope you've enjoyed it. As I say, I'm really pleased that people of your tender young age, Marcus, are interested in science, interested in what's wrong with it, interested in what's right with it, interested in what's true objectively in the universe, and interested in, at a very young age, doing the right thing by yourself.
Yeah.
You know, I had a very many interesting discussions with my not as young as you, but still quite young nephew, who has a particular theological belief structure, which we don't need to go into, but he is religious. And we were talking about some habits that are apparently good for a human body and otherwise. And he was saying that his particular religion teaches, among other things, that it's not your body and you should return it to God in good condition.
Yeah.
And I think that if people just live their lives according to that premise, never mind any assertion to God if you don't want to go that way, you can just say, well, look, at the end of the day, your best prospects for the best length of life, if that's of interest to you, and the best quality of life during that period of time, however long it may be, would seem to be best served by consuming a diet which is conduced, obviously, to your speciation, that being Homo sapiens sapiens.
Yeah.
And to do anything other than that would seem to be Russian roulette.
Yeah. That makes sense. Yeah.
And the other take-home message is that there is no science out there that we can rely on, because we've just shown that, haven't we?
Of nutrition, yeah.
Nutrition science, there ain't no such animal.
Yeah.
Nonsense. It's theology. It's all bought and paid for.
Yeah.
It's garbage. I know I used to teach nutrition science as an academic, believe it or not.
Okay.
Topics. Yeah.
Yeah.
There you go. All right, Marcus, look, it's been an absolute pleasure. Let's do it again. How would you feel, by the way, put you on the spot publicly right here and now, how would you feel about doing a session like what young Lisa is doing, where you come on and we throw the questions at you and you attempt to answer them, and I help out and correct if I need to?
I haven't seen that, but I could do that. I would do that. Yeah.
Basically, all I'm saying is, do you want to be a co-host on a question and answer session where instead of my boring answers, we get your perspective and I just add anything after you've given your answer?
Okay, so like a questionnaire with a Q&A with the chat, basically.
Yeah, so what happens is it's live, it broadcasts live, and the audience asks questions in real time and we answer them live.
Okay, yeah. That would be fun.
All right, book it in on my calendar whenever suits you, really, and we'll just go live at that time, and there'll be people around the world that watch me that go, oh, let's see what Marcus thinks about X, Y, or Z.
Cool.
Be awesome. All right, see you then.
See ya.
Cool. Ciao for now.